Hello everyone, myself Chinesh Kaneria. In this video, we talking about the humoral and cell-mediated immunity. So humoral or cell-mediated immunity are types of the acquired immunity. So first of all, we discuss about the origin of B and T lymphocyte. So B and T lymphocyte are originated from the bone marrow hematopoietic precursor cells, and it will be developed from the bone marrow lymphocyte or RBC, platelets, monocyte or granulocyte. So these are the classification of the bone marrow into the lymphocyte or monocyte, platelets or RBC cells. Bone marrow and lymphocytes are generated into the thymus, are related to the T cell, or lymphocytes are generated the B cell. B cell further divided into the mature form of the B cell and then it will be differentiated into the bone marrow. B cell and T cell are peripheral types of the lymphoid tissues. It's contained into the spleen, Peyer's patches, lymph node or tonsils. And peripheral lymphoid tissues are divided into the mature form of the B cell or T cell, and then it will be exposure to the particular antigenic molecule. Then T cells or B cells are develop the antibody mediated immune responses or T cell mediated immune responses. First of all, we discuss about the humoral antibody mediated immunity. So it will be responsible for the pathogenic components are release the particular antigen, and then the antibody will be interact with the particular antigen. So antibody produced by the lymphocyte can bind to the antigen and marking them for the destruction via phagocytic cell or complement protein. So these B cell are produce the specific types of the antibody against the particular antigen. Humoral responses are developed when antigenic receptors are interact with the particular antigen. Then B cell that are differentiated into the specific types of the B cells. So these specific B cells are generated different memory cells or plasma cells. So these memory cells and plasma cells are interact with the particular antigen, and antibody can be produced against the particular components, and then plasma cells secrete the large amount of the antibody. So whenever the exposure to the particular antigenic molecule, adaptive immune responses are generated, the humoral immunity. So when humoral immunity are developed when the primary responses, initial stage when the antigen will be interact with the particular antigen can bind a particular receptor of the B lymphocyte, and B lymphocyte are further activated and proliferated. Activated form of the B lymphocyte are developed into the plasma cell or memory B cell. So memory cells are also interact with the particular antigen, or plasma cell are secreted the large amount of the antibody molecule. After all, whenever the second response are developed with the same kind of the antigen after the year, then memory cell are further activated, then it will be proliferated or identify the particular antigen, and then it will be develop the large amount of the plasma cell against the same antigen, or further memory cell are also developed. So these secreted antibody are specific for the same kind of the antigen whenever it exposure for at the first time. Development of the B and T lymphocyte. So the development part are carried out from the common lymphoid precursor cell. So these common lymphoid precursor cell and the B lymphocyte are developed into the bone marrow is a generative lymphoid organ. Then it will be transferred or it will be converted into the mature form from the immature B lymphocyte into the blood lymph. After the transfer into the blood lymph, it will be converted into the mature form of the B lymphocyte. or it will be transfer from the peripheral lymphoid organ like lymph node or spleen, and then it will be further recirculated into the bloodstream. In case of the T lymphocyte, so T lymphocyte are developed into the thymus, and then it will be mature into the T lymphocyte into the bloodstream, and then it will be also transfer into the spleen or mucosal cutaneous lymphoid tissues, or then after it will be interact with this lymphoid organ, it will be further recirculated in form of the mature form of the lymphocyte into the bloodstream. So these B cell and T cell are converted into the mature form. It will be transfer into the bloodstream, or then it will be interact with the particular antigenic molecule. Cell mediated immunity. Cell mediated immunity are the lymphocyte is directly attacked to the specific inwards or the antigen by the lysis or inflammation, and it's activating the other lymphocyte or macrophage. So in the condition of the cell mediated immunity, T cell are play important role for the generation of the cell mediated immunity. So the cytotoxic T cell, or is also known as a TC or T helper cell, is directly attack and then kill the cell with the specific antigen, or activated form of the cytotoxic T cell are co-stimulated via T helper cell. So these humoral immunity are developed when it will be interact with the particular antigen, and then it will be activate the B cell. B cell are divided into the plasma cell or memory cell. So secreted antibody that are defend against the extracellular pathogen. So whenever the antigen is engulfed and the display with the phagocytic cell, then T helper cell are activated the B cell, or it also activated the cellular immunity or cell mediated immunity. So these T helper cell are also generate the memory cell against the particular antigenic exposure, or it will be interact with the T cytotoxic cell. So T cytotoxic cell are also directly interact with the infected cell, or it also develop the memory cell or activated form of the cytotoxic cell. So these cell mediated immunity are defense against the infected cell like cancerous or transplant tissue cells. So T cell are developed from the T helper cell are activated the cell mediated immunity as well as humoral immunity. Activation of the T cell and its role into the immunity. So T helper cell play important role for the activation of the both humoral as well as cell mediated immunity. So first of all, a antigen will be enter into the antigen presenting cell will be interact with the particular microbes as a non-self molecule. Then microbes are interact, then macrophage will be process out, and then it will be converted in with the self protein. So after all, the self protein are generated the complex with the non-self complex. After all, a processing or presentation of this particular antigenic molecule, antigen presenting cell are represented at the surface of the particular receptors, and after all, a releasing of this interleukin 1 then stimulate the T helper cell. So after all, a binding with the T helper cell with the antigen presenting cell, the binding site for the antigen, and these are the binding site of particular self protein. So after all binding with these particular antigenic components, T helper cell 1 are also activated itself via releasing of the interleukin 2 via stimulate the cell division process, or it also activated the B cell or cytotoxic T cell. So T helper cell activate the B cell, and B cell are generated the humoral immunity and secretion of the antibody via plasma cell, or after all T helper cell also activate the cytotoxic T cell, and it's also develop the cell mediated immunity, and it will be attack on the infected cell. Antigen antibody complex can signal out the phagocytic cell to attract at the site of antigenic exposure. So first of all, they are the four way of the interactions are carried out with the particular antigenic components. So the binding of the antibody to the particular antigen are inactivate the antigens by neutralization, agglutination, precipitation or activation of the complement system. So first of all, binding with the particular antibody to the antigenic molecule are neutralize the activity of the antigen via block the viral binding site or coat protein of the bacterial. So this bacteria will be coated with the particular antigen or virus will be covered with the particular site with the antibody. Then agglutination condition, agglutination form with the particular antibody via different microbes will be covered with the all specific types of the antibody and the activity of the bacteria will be decreases. The precipitation reaction are also carried out via dissolved form of the antigenic condition and the antigen molecules are interact with the particular specific kind of the antibody and then it will be precipitate out via antigen antibody complex. So these three conditions, neutralization, agglutination or precipitations, can enhance the activity of the phagocytic cell and then this phagocytic cell like macrophage are engulfed for the destruction of this antigen antibody complex. And after all, activation of the complement system are also participate into the antigen antibody complex. Then complement molecule are attached on the infected cell and then it will be leads to the cell lysis. After all, generation of the pores at the infected cell and then it will be lysed with the imbalancing of the osmotic pressure in inner side or outer side of the particular infected cell. The complex are also bind to the surface of the macrophage and to further facilitate for the phagocytic activity. Cytotoxic T cells are bind to the particular infected cell. This self non-self complex are developed with the infected cell. Then after foreign antigen are interact with the cytotoxic T cell and after all the perforin molecules are activated. Perforin make a hole in the infected cell membrane and enzyme can enter through the pores and the hole can be formed and the enzyme that are promote the apoptosis mechanism. And after all, entry of these enzymes are into the infected cell, it will be destroyed via lysis mechanism. Overview of the humoral or cell-mediated immune response. So first of all, antigen or any kind of the foreign protein, bacteria, parasite or fungi are enter through the particular outer side of the environment. That antigen will be digested or it will be represented first of all via cell-mediated immune response. So cell-mediated immune response are developed directly to the antigenic molecule can enter from the outer side the presented via MHC molecule. Class 2 MHC molecule are represented to the T helper cell or class 1 MHC molecule are represented to the T cytotoxic cell. So T cell receptor recognize the antigen can bind to the MHC molecule. After the activation of the T helper cell, the binding antigen with the MHC can activate the T cell. Activated T helper cells are secrete the some kind of the cytokines that contribute to the activation of the B cell, T cell and other phagocytic or macrophage cell. Or T cytotoxic cell are directly responsible to the binding with the antigen and then it will be activate the cytotoxic T lymphocyte or it will be recognize or kill the altered self cells. After the activation of this cell-mediated immune response, T helper cell also activate the B cell. So B cell can interact with the particular antigen and differentiated into the antibody secreting plasma cell. B cell are also responsible directly to the binding with the B cell or converted into the plasma cell. Antibody secreting plasma cell are bind to the particular antibody at the site of the infection, and then it will be facilitate or clearance of the large amount of the antibody secretion against the specific to the antigenic or any bacterial or viral infectious condition. Thank you so much for watching this video. Any query regarding this content you can ask into the comment box. Thank you.